Thanks everyone for coming. I kind of slapped this together, but I read this paper actually a couple months ago, and then I reread it last night, and I realized how much more sense this now, because like I've personally been going down the MolBio rabbit hole for a few months now. I did biomed engineering in school, but like I never really got into like MolBio like very much at all, even despite how incredibly close it is to my professional life. But my professional life is more like, I don't know, mass spec and like chemistry and like analytical chem and stuff like that. But this paper was really cool because, like I said, I got a chance to really apply some of the stuff that I've learned recently with respect to like genes and plasmids and plasmid construction and splicing, and more importantly, like the most important thing was like protein purification schemes. I was looking at this because I had become interested in like that open insulin project. I thought it was pretty dope idea, generally speaking, to like produce insulin. Boo. We got some boos in the chat. I just, simply put, we'll say that I like the idea of us demonstrating that insulin can be manufactured at a low cost, and it doesn't need to be like $700 a month in terms of dosage, in terms of cost for like a dose for like an actual diabetic. I know that there was a team like in Australia doing this, and they had actually like produced insulin and then purified it using a HisTag system, but that is extremely, well, not extreme, but it's like pretty expensive to use like a HisTag purification protocol. And they were looking at other options, and I happened to stumble across this when I was doing research into like my cell-free biology stuff, and I found this stuff really interesting because what they're doing is they're using cellulose as a purification medium, so that like they can actually produce proteins and use cellulose to like purify to elute out a protein of interest, and they do that in this paper. So that's what this paper is looking at. So yeah, let's get into it. So what they did is they tried to find this low cost method for purification, and they did it using the CBM3 affinity tag. And so they used the CBM3 gene from this Clostridium thermocellum, and they used it. They optimized this gene for Pichia pastoris, a particular yeast that they're using in this method to like produce this protein of interest. So they took that CBM3 tag, which is the tag that you put on the end of like a protein of interest. And this CBM3 tag is what actually binds to the cellulose and keeps it bound in that matrix. And then what they did, to actually test this theory about how well this works is they fused the CBM3 tag to this EGFP protein, green fluorescent protein, such that they could express this tag protein like thing in yeast, and then take that cell, lysate that supernatant, and then put it through this purification protocol, and then out comes like just the protein of interest. So they did a number of different variants with the CBM3 tag in order to try to like see what kinds of effects would be relevant when you're talking about this kind of purification. So they did CBM3 EGFP with glycosylations. There was a couple of other variants in here where they had one with one glycan, one with no glycan, and one with up to 3 glycans. Although I don't think that they actually necessarily had all of that many in there. But so they had a few different glycans to see if glycosylation would have an impact on this adsorption capacity. And I'll show you the results at the end. Then yeah, they had so basically yeah, they had a few different mutants that they made. The one that was really interesting to me though was this one, the CBM3 intine EGFP. I'm not even like 1,000,000% sure about what an intine is, but it's like basically a protein that kind of exists in for a period of time. So like in the way that they're using it here, they're binding this CBM3 to an intine and then also to EGFP. And the way that this works is they get this whole molecule, because this is one like molecule, okay? The CBM3 intine EGFP. They get this into the column, and then they run this other reagent through that column, and then out comes just the EGFP. So that way, the intine is kind of like this thing that you can cut from like the molecule of interest. So that way, like the CBM3 binds against the cellulose, and then everything is in like the cellulose matrix. You cut away the intine using this other. They use like DTT and something else. We'll get into it. Oh yeah, L-cysteine. It's right here. And then yeah, you cut the intine, and then all you're left with is the EGFP in the buffer, and that's what flows out. And like I said, the results they show at the end of this are like pretty good. At least for like anything we're trying to get up to in like a hacker kind of environment. And so yeah, that was the cool part about this. That they went through.
So pause here just for a moment, just because I'm trying to focus on having journal clubs be more participatory, in as much as that you're not just like letting the science bludgeon you. Are you guys grasping what's going on here? Like, are there questions?
Somewhat.
And there are some questions that might be like too complex, but if anyone needs a little shoring up, now is the time.
What's meant by a tag?
A tag, yeah, good question. So a tag is like this little. It's like literally at the end of the molecule of interest. So a lot of cases, the people use these in protein purification schemes. So I like antibodies, so I'll just talk about antibodies. So like for example, let's say you're making an antibody. At the end of an antibody, for example, you might attach a tag. Like a common one is called his tag, which is like literally just a bunch of histidines like in a row. And then what you'll do is you'll have a matrix that binds really well to like histidines, and then that tag will like anchor like that antibody against like your matrix. So that way, it's like the tag is like physically what's like holding on to it. It's like what's a good example? Like literally an anchor. Yeah, like I'm really just gonna go with that. Like, so let's say like you got your boat, and like your boat's flowing around, but like you throw down the anchor, and like that's obviously gonna keep it stationary wherever it is. Similarly, that's what this tag is doing. Like in the absence of the tag, the molecule will just kind of like flow through this matrix, and then like you won't be able to capture it. But with the tag, like this thing really acts like an anchor, and it'll just sit in the matrix and until you do something to it. Yeah, does that make sense?
Yeah, thanks.
Okay, cool. Very good.
I have
anything else?
Yes. What's glycosylation? I don't know if it's too off topic.
You're
Yeah,
really
for sure.
quiet, by the way.
It is quiet, but I did hear you.
Oh. Is it better? I don't know. Yeah.
That's better. Yeah. Oscillation. I want to see if I can just. Okay, I want to. Yeah, you guys should be able to see this now. Okay, a glycosylation is like a sugar basically. It's a glycan. It's also known as a glycan. I'll click on this one. So again, antibody stuff is like my jam, so I'll talk about that. It's literally like a sugar molecule on the side of like your in this case an antibody of interest. And so glycans can do a lot of stuff. So like they can be they're expressed naturally in antibodies. Like you really need to have glycans on an antibody in order to get like the proper folding, and in order to get like usually the proper like reactions depending on like what you're doing with the antibody. But for example, yeah, like there's a lot of things that like the glycan gets involved in terms of like the actual like chemistry, I guess, behind like an antibody, for example. And so in this case as well, another one, it's easy to look at. Spike protein has a lot of glycosylations as well. And so yeah, like lots of like proteins, lots of molecules in nature like have like glycosylations. So these like sugar molecules that are like attached to them or made in them or something like that. So like membrane glycoprotein, that's kind of what we're talking about as well. So yeah, it's just a part of the molecule. It's another like piece of the molecule that's there, and I can talk a lot about it, but that's the kind of like the short answer. Does that make sense?
So it's like a sticky bit.
Yeah, it can be sticky. And in this case, I guess it's actually not sticky. It doesn't bind
Okay.
too well to the cellulose, but yeah, it's a part of the molecule. It's like a piece of it.
I might Google it later then.
Sure, yeah. I'm no expert, but like I know that
Yeah,
it's there.
thank you.
It's
Anyway,
it's relevant. And yeah, it's basically a sugar. Yeah.
I have a
thank you.
question.
Go for it.
Is I'm trying to figure out why they would focus on the inteins. Is it because those are the proteins that they want to extract and purify? Because those are like almost excised proteins from protein splicing.
Yeah, the use of the intine is that you can use it as we're reading on the wiki.
Okay.
Is that you're able to excise itself and join the remaining portions. The exteins in this case would be like our EGFP, I guess, and CBM3
I see.
with a peptide bond during protein splicing. So, yeah, the idea here is that you have this like intine, which is like linking two molecules of interest that we can just like chop off and then allow the other molecules to like stay intact. That's the
Gotcha.
point of it. Like, because there's a number of ways you can purify something. Inteins have been rather popular, I think, in the past like couple decades, especially from what I've like read about them. Because you can do this kind of chemistry where it's like you have a molecule of interest, in this case like the CBM3 and EGFP, and then in the middle you put in this intine, and then the CBM3 stays bound to the cellulose matrix, and that's cool. That's what you want. The anchor is anchored. But then you chop off the intine, and now like this EGFP is like free to flow down the column and be used subsequently, like once it's outside of the column.
I
So
see.
in this case, yeah, the intine is like this. It's like the rope between your
Gotcha.
anchor and your boat. And we're gonna use some scissors in a bit, and that's what lets us grab the protein out. Yeah,
Cool.
cool beans. Yeah, is there anything else? Okay, cool. Yeah, so like I was saying, the intro behind this I kind of gave. I mean, I don't need to spend too much time. I think going over it, but like basically the premise is there. We want to use this cellulose stuff because as they point out here, at less than $5 per gram, this regenerated amorphous cellulose RAC is really inexpensive. I actually don't know what a comparative cost is between like this and like a Histag system, but I know that it's like a lot more money. I've just never ran with that kind of stuff personally, so I really don't know the exact cost. I don't know if anyone else knows what that cost might be, but nonetheless, yeah, this was really appealing system just because of that raw cost, like $5 per gram. I mean, like, listen, there's very few biological agents that I know of, generally speaking, that are $5 per gram. Like, I know that antibodies are something like several hundreds of dollars per like 100 micrograms or 50 micrograms.
Yeah, I was about to say like a mic. Yeah,
which is like
I don't know. LB is cheap, right?
yeah,
but that's about it.
Yeah, that's about it. So I mean, yeah, like if you're trying to get like a gram of like an antibody, it costs you like thousands of dollars. And so like Histag reagents, I think similarly are like, expensive. So RAC is really cheap, which is great. Also, its properties are highly amenable to protein purification. For example, it can withstand really high centrifugation speeds, like 12,000 Gs. Like that's really good because we're going to be spinning this bad boy, so it needs to be able to live through like the spinning that we're going to be doing. Cellulose is inert, which is great, doesn't react with other things. Has low non-specific affinity for most proteins, which is great because it doesn't like bind to other things that it's not supposed to. It just binds to the CBM3 tag that we have in there. And yeah, there's a lot of different versions of it, like including cotton membranes, powder, fibers, and beads. So there's a lot of different ways you can work with this kind of stuff, but they chose one, and at the very end of this paper, they give the rationale of like why they specifically chose RAC over like a number of these different things. And I think that like the rationale it makes sense, and I would probably go with RAC as well if I was well, I should say like when I actually like legitimately want to use this kind of like purification protocol once I work my way up there. But yeah. So yeah, they talk about how they've demonstrated this in a number of existing systems. Like people have done this exact kind of like protocol on E. coli. They wanted to specifically show that it would work in Pichia pastoris. And I think I was looking at Pichia pastoris because it was working really well for like some proteins, some therapeutic proteins that I was looking at doing in cell-free applications as well. So naturally, like this seems like a really legit system. Yeah, to use this kind of protocol on. Call the application, blah blah blah, yeast not reported. They talk about the inteins. Okay, yeah. They optimize this purification. They optimize the codons for this particular tag in Pichia pastoris, which is good. So we know that like the codons are optimized for this yeast. And they investigated the N-glycosylation effect and absorption capacity, like I said earlier, by creating multiple variants, multiple mutants to show, what the effects of the glycan might have on this binding activity. Cool. Yeah, I think it's worth reading all of this, but yeah, it's basically them. A lot of this comes down to like them saying, like, we've tried doing this in a few other systems. It worked well. We wanted to specifically mention, like, or demonstrate how this works in like this Pichia pastoris kind of like context. Yeah. Any other
Excuse
questions?
me.
Yeah, go for it.
Yeah. Justin, I think his Spider Bear project is using Pichia pastoris.
Oh, cool. Yep.
That is correct.
Justin's Spider Beer. I've never met or talked to Justin. I just watch his videos as well. But that's really cool to know. I didn't. How did he do the purification? Right? Didn't he have like a supernade and like he crashed out a bunch of stuff?
Yeah, he just it just crashes really easy actually.
Yeah.
By using a specific solvent, and then everything crashes out, and then you just rehydrate it, and it turns back into liquid, and then you can literally just like load it into a syringe and pipe it in, and it turns into strands. It's actually
Really?
very simple. Yeah. It's,
Huh.
Yeah, part of it has to do with the end group, but basically the exposure to water causes it to turn into a strand.
I was wondering, how it would strand up. That's really cool though. So I guess like because it's because he's adding water, but is it also because he's putting it like through a channel?
Yeah, he just squeezes it out through a 5 milliliter syringe.
Cool.
Yeah, it's kind of been a pause on the project just because he's he's always got like another thing going on. So
yeah.
But yeah, we have some of that here now. So I'm going to just try to make it into sheets. Anywho, yes, that is the yeast that Justin is using as well.
Cool. Good to relate that. Cool. Materials, methods, chemicals, and strains. They talk about everything that they used. I recommend reading this, but I'm not going to spend a whole lot of time doing it. Other than to highlight, I was doing this last night actually, which is this. So they mentioned the specific kinds of cellulose that they picked up. You have your LB media, Pichia pastoris. They mentioned the plasmid that they were using as well. They mentioned the different kinds of mediums. And Pichia pastoris. I'm just sorry, guys. I'm actually looking at I underlined some stuff, but I underlined quite a bit last night, I guess. So I'm just trying to highlight some of that stuff to you guys as well. And here's your other plasmid. Yeah, here's the same plasmid that they had used. To make this one. Okay, construction of the expression plasmids. So this was the stuff that I had a lot of fun reading over again last night because there was a lot of like fusion stuff going on here, and so here's actually a bunch of primers that they used to like amplify the genes of interest when they were doing the splicing. So you can go through and like follow like all of this like stuff because they detail it quite well. But basically what they're doing is they're doing amplification of the region of interest, and using restriction enzymes and restriction sites to copy and paste in specific like genes, like encoding for like the stuff we're looking at, like the CBM3 gene in that they've bought from like other plasmids. So like they bought this CBM, or they have the CBM3 gene. It was subcloned using these restriction enzymes. Sites, using these restriction sites, and then they because they had it in this plasmid, and then so this is
so
if
people don't know about restriction sites, restriction enzymes, stuff like that, that's why you should jump down to the Plasmid Creation Club and listen to those guys talk, because that's the type of stuff that they're starting to work on, if I'm not incorrect here. Just trying to get everyone on the same page.
I haven't watched the last one. Yeah, no, I agree. I haven't watched the last one, but I feel like
they're getting there. They're getting there.
If you want to quickly look at what it is, like here's a quick picture. I don't even know if this will show up that well on my screen.
Everybody loves that picture of the bacteria with the plasmids in it.
Yeah.
Yeah. Like a freaking Mike and Ike's for genes.
Oh, stop it.
Actually, I think you can see the restriction site that they used. EcoR1. Yeah, there it is. EcoR1. It's here as well. And so what these are just like spots where you can like design your insertion kind of like scheme. And that's what they did here. Is like they picked a specific spot in this plasma where they were like, okay, we're gonna chop here and here. And these were like on flanking ends of like the gene of interest on either side. And then they can amplify just like this gene of interest, including these restriction sites in this plasmid, so that you have a bunch of like this gene that encodes for CBM3. Now a bunch of that stuff's floating around, and then they did the same thing with the EGFP. So now we have a bunch of EGFP like gene floating around with these restriction sites that like will mate, and then it's just kind of like Legos, right? Like you're getting things to kind of like fit nicely into each other, and then at the end of it, you're gonna have a plasmid that's gonna code for CBM3 and EGFP. And that's what they were doing here. So they have the primers and stuff that they made like listed. And this is stuff that I found really fun last night. Like I said, it was like, oh cool, like I actually get what's going on now. We're building a longer gene that's going to encode for CBM3 and EGFP. And then they kind of just did more of the same as we go through here. They have a few mutants that described, like I said, with the glycosylations and not. And as we go down, like again, more of the same. They're talking about like this stuff. This is, I think, where they're getting into the INTEEN protocol. Yeah, EGFP INTEEN CBM3 protocol is here. Again, more of the same, where they're amplifying that gene of interest, splicing them together, and then making a plasmid that actually has like this entire fusion gene in it, in the p-ship Astoris expression plasmid. And then they made another one down here with inulinase. Really not familiar with that. I was like I said earlier, really specifically focused on like the INTEEN stuff because that looked the coolest to me. But I think this inulinase stuff is also another way to do that purification. Yeah, cool. Before I get into the mutants, are there any questions about that? Okie doke. You want to learn a little bit more about the glycans? Like here's a quick, kind of like footnotes about it. Glycosylation is the most common post-translational modification, PTM, of proteins secreted in eukaryotic cells. They occur at this kind of motif, so it's asparagine something, and then a serine or threonine sequence motif. That's for N-linked, and you see that in antibodies. Like there's always that always exists in antibodies. that are like full length, that are like an IgG, that have a constant region. But also sometimes we'll just kind of like randomly see them as O-linked glycans that are attached to like either serine or threonine residues. In this case, this molecule, this CBM3, has 3 asparagines at 14, at 68, and at 124. And so they tried to like basically create mutants that either like just had the 1 or like 3 or none, and that's what they did. They actually made a number of different constructs, but they only actually worked with like these 3 because they worked right away, and they're like, okay, like, well, we're not doing the rest. And that was good enough. And so they named those the wild type. So WT for wild type. This is like with the 3 potential N-glycosylation sites. This CBM3 mutant, first one has no glycans, and the second mutant has this glycan site at position 68. And yeah, and so that's how those are the 3 mutants that they made.
Can you break this down to me like it's a Lego kit?
Yes, I think. I mean, so basically you have like the maybe this makes more sense for actually like the picture. CBM3 molecule.
So we got the WT, the MT. We've got some sort of N68. Like, talk to me in English, man.
Got you. I want to find a picture of this molecule so that this makes more sense. Well, okay. Basically, what we're saying here is that there's 3 potential spots at which 3 asparagine residues at which like we could have a glycosylation like attached to the CBM3 molecule. Okay. So this wild type 1 has all 3 of those sites. It has all 3 of those asparagines in it. And so it fits that motif that we just described above here, where it's asparagine, XXX, and then serine, threonine. Like there's 3 of those sites naturally occurring in the CBM3 molecule. And then in this mutant, this first mutant that they made, they eliminated all of those. Like they removed that motif so that there should not be any glycosylation happening on the CBM3 molecule that they express in the Pichia pastoris. The second mutant has 1 asparagine site, and it is here between well, Q, but like I'm forgetting I'm forgetting what Q is. That's not super important, but it'd be really cool if we could just quickly mention. But I'm almost there. It's glutamine, yeah. So between the asparagine and then the glutamine, this is position 68. We have this one motif which satisfies our criteria above, which says that we can have a glycosylation site expressed here at this spot. And like I said earlier, like the reason they did it this way is they have like the one with 3 glycans, the one with no glycans, the one with 1 glycan. The reason they did that is because they wanted to see if the glycan would impact binding of the CBM3 molecule and the cellulose like matrix that we push all of this into. And then I'll show you the results at the end, but like it didn't really affect things too much. But they wanted to find that out. They wanted to be sure about whether or not that would affect things. Because like I said earlier, glycans can really matter. They can really influence a lot of chemical reactions, a lot of biological reactions. And so they thought that, it'd be worth asking the question here and not too much more work to find out the answer by just, splicing in some variants that don't have the asparagine residues. Yeah. Okay. Expression of the CBM3 fused protein. So they had expression plasmids, linearized. This is getting back into the plasmid club stuff. I don't know if they've gone this far into it either, but one thing that you do when you try to like splice things together is you try to like linearize them so that they're open up again and you can actually splice things together. Because if it's just a circle, like plasmids are circular pieces of like DNA, and they're harder to like work like to splice things into if they are still in a circle. And so what you will do is you will make it into a strand. Like you'll literally chop up some of it using the restriction enzymes we mentioned earlier at the restriction sites, and that'll chop up this like circular thing into like a strand, and then you can splice in your genes of interest together. And the way that the restriction sites will work is that they'll come back together. They'll eventually be like, oh look, it's our friend, you know, that binds well here. And this is, I think, tricky to kind of like show without pictures. Let me see.
The term for it is a sorry to interject. It's
Go for
the
it.
cloning method, and we have like multiple different ones. Some of them are super patented, some of them are super unclear. And like for example, Gibson assembly is the most common one. Then you have
I'm gonna show you guys some stuff, but go ahead, continue.
yeah. Then you have like another one which is, I don't know if it's JPL should have been there. He would, those things just flow out of him like it was his second language. But yeah, as far as I understand it, it's the restriction cloning is heavily patented, and yeah, that's all I can say. But that's how you join together those sticky bits, or you cut them apart and
Okay.
you like you shove
I have
extra
pictures.
bits together.
So I also recommend this series of lectures. Like some of you guys might know, like I'm pretty close to the University of Toronto, and naturally I'm trying to get in with a lot of these guys, just because like they're friendly and like they'll talk to me. And there's a series of videos from this thing called CS Berg, and they talk a lot about this stuff. Like this guy's name's Patrick. He's a nice fella, but he talks a lot about this kind of like stuff. And you can go watch his lectures as well. Like they're available on the internet
Yeah.
now with CS Berg.
Would you link it to the Plasmid Club? I would highly appreciate it. If like
Yeah, for sure.
afterwards. Cool.
Yeah.
Awesome.
Yeah, absolutely. Yeah. Not to spend too much time on it, but I really like this guy. Like the way that he teaches is very good. He knows a lot about it. He's a PhD and he's doing his own stuff as well. But yeah, he's been really really kind in sharing some information and putting this stuff up for free. So anyway, to our point about this insertion of genes of interest and the plasmid. So like you have this construct, you want to insert, like the green ends go together, the blue ends go together. That's what we're doing here. This whole circular thing is your plasmid. This puck, 19 is your plasmid, but you're going to insert a specific gene of interest. Use these restriction sites. They act like these kind of color-coded Legos, so that you can just kind of like pick them, as you can see here. Chop off the bits that you don't want and insert the bits that you do want. Yeah, 3 inserts, green to green, blue to blue, orange and reds. This all, I think, makes things way easier to understand instead of me just rambling on about how these things act. So yeah, basically this is the idea: is you use these restriction enzymes and these restriction sites to create a mating of pieces of DNA. So they just plug together. They just kind of like work together because like this, recognizes like recognizes like, and the reason is because they're actual DNA like code, like the actual sequence that you're using mates. So like T goes to A and A goes to T and G to C and so on.
Right.
And so we actually create these overhangs such that in this case, the NDE1 cut site, like here it'll cleave this overhang, and you have like a TA overhang. Then when the gene of interest that you want to mate like also has this overhang that matches perfectly, like it'll go together by like affinity, because that's like naturally what should happen there. Like I don't think it's always so straightforward as like a two nucleotide site like this, but like certainly it can and work like that. So anyway, yeah, that's a little bit about like what we're doing here with like splicing these things together. They work in
Cool.
this.
Yeah, it was the Golden Gate Assembly, the word I was looking for. This is like the Golden Gate Assembly is the super unclear one. Only like very few person uses it, but it should be like, yeah, awesome.
Yes.
Awesome. I can't wait to read on it.
Yeah, for sure. Yeah, these guys are really cool. So I will I'll post that stuff in the chat a little bit later on. But that's the idea behind what we're doing here as well. Is we're splicing a bunch of these genes together such that we now have like a gene that codes for like each molecule of interest, and now we splice them together. So that's what we get: the CBM3 fused with the intine, fused with the EGFP, all inside of this P. schistosaurus expression plasmid. And yeah, so that's what we did here. Is they did this. They had the plasmid ready, and then they started feeding it the juice. They gave it the culture media, and they incubated it on plates. And then yeah, they did this for a few days. Erlenmeyer flasks came back, centrifuged it. Actually, one question that I do have about this, because something that I've never done before, was they mentioned like doing here. They mentioned it a little bit. Like they did the centrifugation. Yeah, the cells were recovered by centrifugation at 2000 G for 10 minutes at room temp. Oh, this is not what I wanted. No, this one. Yeah. The selected strains were inoculated in 500 mils of this medium in a 1 liter Erlenmeyer flask, cultivated at 30 C and 250 RPM for 24 hours. So like if you're gonna do this, like I have an incubator in my community lab as well, but like would you literally put what is the word? Centrifuger?
Shaker table.
Centrifuge? Centrifuge.
Shaker table. Shaker table.
Shaker table. Okay, that's what you do.
Yeah, you just put a shaker table.
And it'd be like battery operated, I guess.
No, you just run a cable out through the incubator.
Oh, okay. I was curious about. So I guess like, do incubators have like a spot to do that normally?
Some of them do, but you don't necessarily need it because most of your doors have like a flexible seal, and they will still work. You can totally run a cable out through it.
Okay.
Put a shaker table in the bottom and just shake it.
Shake it.
Yeah.
Okay. Cool.
Mm-hmm.
I gotta make sure
you
we
actually
have
the ones that have the built-in shaker table. That's another thing.
Right. Yeah, I don't know that ours is gonna have that. We we got some like I think it was Sanofi Pester was like throwing some of these out, and like we got a couple in our hands. So we have one that like actually works legit. Then we tore apart the other one because we're making a flow hood out of it. And yeah, we're just waiting on some of the supplies to come in to finish that project. But yeah, and we also I saw one shaker table that like we have in our lab, and it is literally like a floppy disk repurposed. Like someone hacked together like a floppy disk to like be a shaker table.
Like a disk drive, like an old like floppy disk reader.
Yeah,
That's amazing.
it literally. I should send you guys the video. Yeah, I took a little video of it. I found it because I was like I just got into this lab a month ago or so, and I was pilfering through all of like the equipment because I want to see what we had on our hands, and I was looking at. I was like, what is this little box? And it's like a little shaker table. I'm like, oh my god, it's a floppy disk, and like someone just hacked it, and like they plugged it together, and it works. It works. Like, and you can adjust the speed. You can adjust the stroke length. It's kind of funny. Yeah, how well it works. But anyway, I was curious about that, but that's good to know. So yeah, so they incubate. They get these yeast. They culture the yeast, and then like it'll produce the protein of interest because the cells will multiply with the plasmid inserted, and like thus they're gonna multiply and produce the protein of interest inside of the cells. And then once they were done, they harvested them by centrifugation at 4 degrees. Another question. So they took the cells, they took the supernatant, and then they centrifuged it at 4 degrees. Like, how would you do that? Like, would you put a centrifuge in a fridge?
Yes.
Refrigerated centrifuges are a thing.
Okay, so you would
there's a centrifuge that has its own cooling unit. Also. Gotcha. In a lot of professional labs or university labs or industry labs or however you want to call it, you basically have a walk-in fridge and you do your lab work in there because then you never have to worry about ice blocks or any other stuff like that. You're just always working at 4 degrees because you're in a refrigerator. So this is a very standard type of thing, but refrigerated centrifuge is totally a normal thing. That's how that happens.
Gotcha. Yeah, I guess so. In the absence of said centrifuge, I would probably try to stick a centrifuge in the fridge.
Indeed. Yes.
Yeah. Okay. That's probably what I'll end up doing. But okay, cool. That's good to know.
Really not that. This is like one of those steps where you're going for perfection as opposed to just doable.
Yep.
If you want the optimal conditions, you have a refrigerated centrifuge or you're in a walk-in. But you could probably do this at room temperature as long as you're just rapid and keep your materials on ice.
Gotcha. Okay. Cool. No, that makes sense. Thanks for the insight. Cool. So yeah, they took the cells that they harvested, they centrifuged them at 4C, then they washed it onto the cell lysis buffer, which is this string of compounds, and then they resuspended the cells in 30 mils of cell lysis buffers. And then they crushed them in an ice bath and by ultrasonication. So they and they did these specs on it, and so then they had the cell lysate, and the cell lysate, which is now like the shredded like components of like the cells and like what's inside the cells, which will include our proteins of interest with a tag on them. That's what we were working with here. And then they put that through the purification scheme. And they did two different variants. So they first pass, so they added the this is the cellulose and the supernatant. So they added this all like just together in one big mash. And then they had varying amounts. And the other iteration path was that they just added the supernatant to the RAC column, which you're gonna see pictures of in a second, and that'll make more sense. I just want to flip through here, make sure I didn't miss anything else that was particularly interesting. They talk about like why they mix certain amounts and stuff like that in here. They use ethyl glycol in order to do some of these elutions, which is really good as well. I think this is for the. I'm actually not 100% sure. So it'd be worth confirming. But I think that in one iteration of this, the inulinase 1, they actually wash out the protein of interest with ethyl glycol, and that kind of acts similarly to the intine process that we'll show you in a sec here. Basically they take this stuff, and then the elution was accomplished by mixing thoroughly with EG and RAC and centrifuging the column in a larger tube that is compatible with the centrifuge motor. I'm actually just going to show you guys this picture so this makes sense. So like they literally add the supernatant, the yellow stuff, into this column. In this case, this is the white stuff at the bottom is the RAC. This is your cellulose. They add the supernatant on. They do some mixing with some chemicals in order to try to get everything to go bind into the cellulose column. And once we have that kind of thing figured out, like most of your stuff is actually in the cellulose, the protein of interest is in the cellulose. Then they put this tube, this column, into a larger Falcon tube, I think is what they used, and then they literally just used they centrifuged that, and then out of that column, they had this other like PCR tube, this little tube sitting at the bottom to catch the protein of interest. And in this case, as you can see, like that is that's your EGFP right there. That's your green fluorescent protein in that little PCR tube, and it looks good. And they actually showed that worked really well. So as we're like thinking through like this whole thing, that's basically what happened. The centrifugation speed was dependent on the tolerance of both the column and the tube. They settled on this 5,000 Gs, and then they ended up basically going through this kind of experimental protocol. So that was for the inulase one, I should say, but they did the same thing for the intine one. They just had different buffers and stuff that they washed in like this kind of like fourth and fifth step in order to get the chemistry to work correctly. And then they measured adsorption of this thing on the RAC, and so a few different ways in which they would measure the absorption. But basically, they did like a fluorescence detection and protein analysis. So they actually used fluorescent intensity excitation at these wavelengths using a SpectrumX microplate reader. You can go and get one if you can get your hands on one. I'm building a spec using some really accessible stuff. So I don't know that you need like all this kind of jazz. I spend a lot of money on these kinds of things, but we could talk about that later. But like, the point of this is, they were trying to measure the success of this by measuring the fluorescence at each stage of the purification, like the supernatant, getting a reading off of that, and then getting a reading like post purification and stuff like that to try to get a quant of like how much of this stuff actually eluted, and it seems to have worked reasonably well. Then they kind of like went back and quantified that against a Coomassie blue gel stain in this picture here. Coomassie blue is a really nice gel to use because it is, relatively speaking, quantitative against proteins. So the darker a band stains on Coomassie blue, the more protein that there is, generally speaking, which is nice because you want to try to get like a rough kind of protein estimate. This is how people do it in industry. At my professional setting, like this is how we do it. Like when we receive an antibody in our hands at work, like we always run a gel first, and that gives us an idea of like, A, is it just an antibody or are there other proteins there? And B, like how much is actually in there, because we try to figure out, if there's enough antibody for us to do our analysis on or not. Yeah. Okay. Cool. So this is kind of like, like I said, more proof of what's in the pudding. Trying to show you that there actually is a protein of interest and how they quantified it and stuff like that. And then trying to measure the absorption, trying to see how tightly this stuff binds. And then in these kinds of like sections are really just talking about like, we saw this kind of binding activity with respect to the protein of interest and different concentrations. So like cleavage ratio. They wanted to see like, like how much of this protein did we actually get in there, and then B, like how much of it was eluted and came out the other end. And here they had different concentrations for like the reagents that they were using in order to try to assess like which ratio or which amount of this reagent would be like the best in order to achieve like higher levels of cleavage. And at different temperatures as well. They try to see if temperature would have an effect on the cleavage ratio. As you can see, higher temps work better because they speed up the reaction. And in this case, there's not a lot of difference between 23 and 37 degrees. So like I'd probably run with like something like this if I was going to do it. 24 hours. Yeah, 24 hours. You have about 70-ish% cleavage at, I'm sure, actually I forget how they standard which concentration or which molar concentration of the DDT they standardize this one against, but that's probably something that I would run with. But you can always go for longer and get higher yields as well. As you can see, like they had a higher yield at this concentration after 50 hours, like almost 2 days, over 2 days. Cool. And this is more of the same data, I think. So this above one, self cleavage of GIC with DDT as the inducer, and this is self cleavage of GIC with L-cysteine as the inducer. So this is for the intine case that we were mentioning earlier. Yeah. Okay. We're almost done. But again, more of like confirming stuff, like getting into the ratios, like what cleavage levels were achieved on what mutants or what strains, and like temperature having an effect. It's just read the graphs, and this is kind of like what they're talking about at this point. Before I go on, I guess like I'll ask, are there any questions about any of this so far? Okay. And then a discussion. I think it's just kind of like more of like the same stuff that we've really been talking about. There's nothing like particularly like novel or like new to like extract from here. Intein's been widely used to remove affinity tags following protein purification because they're nice. This is the one that they used. Oh wait, no. this is another one, but I forget if this is the exact one that they use in this case or not. I think they use a VMA one. Yeah, this is the one. Yeah, this one they use actually because it doesn't actually doesn't self cleave with in vivo conditions. That's something that Inteens will sometimes do, is like they will bind like a protein to another protein of interest or something like this, and then before like a protein even comes out of the cells, for example, like it'll already be cleaved off, and then like you'll only get like the protein of interest as like the end product. In this case, they wanted to use this Inteen for the same reason, but they wanted to choose this specific Inteen because it doesn't tend to self-cleave very much in vivo, like in the yeast that we're looking at, which is nice. So it actually worked well because then we could cleave it like when we told it to, when we ran it through our column and like use the inducer, the DDT or the L-cysteine. Okay, last page here. Many impure proteins remained on the RAC following the wash step, which is good. That means like other things got caught in that matrix, but not our protein of interest. And they said they were irreversibly absorbed to the RAC and can only be removed by boiling with SDS solution and like shredding these things apart, presumably. Like obviously the other proteins, if they're getting boiled, like aren't probably going to stay in their conformation. They're probably going to like denature and like not be the same shape that they were when they went in. So this is really good news because basically everything bound really tightly to the RAC, didn't let much through. The only thing it let through in this case was the EGFP in its buffer, and then everything else was stuck in there. And yeah, this system worked really well. So they have demonstrated that we have a very low concentrations, that even at very low concentrations, the target protein could be purified. Notably, 60 to 80% of the target protein could still be absorbed to RAC. In the inulinase case, the supernatant was purified at a low concentration. Approximately 74% of this inulinase variant was adsorbed, which is nice. So, like most of it stuck to the RAC, was anchored in there. And so now we can suggest that the CBM3 is actually a pretty good high efficiency affinity tag to be used in yeast systems. Yeah, and I think that's pretty much it. That's the main finding here, is that it worked. It worked pretty good. They talk about some other like things you can do to improve it. You can wash it like multiple times. Like, where is it? 2 to 3 elutions are recommended to further improve the purification. Good to know. We'll keep that in mind as I do this.
So I have a question.
Yep.
So what are the and if you could, because I know that you're all about the antibodies, but if you could not be exceptionally about the antibodies, what are like so what's this good for? Like, why do I care about this?
Yeah, for sure. It's a good question. So, like, let's say you're a biohacker and you want to make a protein of interest, any protein, generally speaking.
Like something that makes plants glow?
Sure, absolutely.
Sorry, I just sorry.
We gotta plug in our favorites, absolutely. The whole point of this is like, let's say you want to use a protein of interest. Like, so you might want to make a protein because it's hella expensive on the free market. Like, if you want to pay someone to sell you like a protein of interest because you're using it for like some experiments, like that might be pretty expensive. You might say to yourself, well, I could do this in house. I could make this stuff for like 1/10 or 1/20th the cost. Like someone's gonna sell it to me. And then you say to yourself, okay, I could do the gene construction, I could do the plasmid construction, I can make the protein in these cells, and then you have to ask yourself, like, how do I get just the protein out? And like that's what this paper comes in, and this technique comes in, because like if you have like the protein you want like inside of cells, like that's cool and like good job, but like you're not going to be able to use that because you can't like take this slurry of yeast like stuff and like get the same effect out of it that you would if you just had the protein of interest. And so that's why we're using this kind of purification technique, is because we just want this one protein of interest. And this paper obviously focuses on EGFP, but like, you know, one can extend this further and think of like other proteins that you might want. You guys know I could just sit here and talk about antibodies, which is like fine, but we're trying not to. And so, you know, whatever other proteins of interest might be relevant here, you know, similarly, you could tag with this CBM3 molecule, put it through this RAC column. Where's the picture? Yeah, put it through this RAC column, and then just that CBM tag acts as an anchor, and then, you know, cut off that rope using that inducer, and then outflows just that protein of interest in the buffer, and, you know, that's how you get a good amount of that protein. In this case, like, I think what they actually ended up producing was like a few megs of EGFP. I have to actually look at here again. How much did they actually get out? They had titrations down here. Yeah, 50 micrograms per milliliter in 2 mg per mil impure proteins. This is a really low concentration that they ended up actually pulling out. Like, there's 1,000 micrograms per mg, so this is 50 over 2,000. Yeah, and so that's like, what's that, like 5, like point, very small percentage, 50, 0.025%. No, 0.025 times 100. It's, yeah, 2.5%. So 2.5% of this entire mixture was just the protein of interest. So you guys can imagine that, like, if 97.5% of, like, you know, what you're getting out of like the yeast in terms of just the proteins is the wrong stuff, like, you're gonna have the wrong reaction. So yeah, getting a nice concentrated, highly pure CBM3 tagged, like, molecule is going to be really useful. So hopefully, yeah, that makes sense. Answers the question.
Yeah, that worked for me.
Cool.
Anyone else? Anyone?
Maybe.
Okay.
Did I understand this paper to say essentially the amount of cleavage is dependent on the temperature?
Some of it is, sure.
I think I knew that from my teen years, but okay.
Yeah, for sure. Yeah, temperature always plays an effect, right? That's your science 101 stuff, right? Like, yeah, temperature.
You're too sciencey.
Sorry, I can.
No, the cleavage. Come on, cleavage.
I'm looking at cleavage right now.
Oh, I see what you did there.
Oh my god. Yeah.
Your focus is so narrow right now.
Yeah, you're right. I should expand my horizons. I should.
You need to take a break, man.
Yeah, where's my girlfriend?
Go look at some proper cleavage. There you go.
Oh boy. Oh boy. Well, we've got some cleavage here for sure. Temperature dependent. Let me turn up the heat in here.
Temperature dependent cleavage. Mm.
Oh jeez. Okay.
Pretty hot.
All right, nerds.
Yep.
Do we have any real questions? And then there was
No, I
absolute
loved
silence.
it. I enjoyed the hell out of this show.
Very good.
Cool. Well, another successful,
Oh,
though oddly timed, but it happened. Good job.
It happened.
And thanks everyone for sticking around.
hold on a second. I did have one other thing. When you were saying you wanted a picture of this, of the protein, isn't that what you were saying?
Oh boy. Okay.
I should have asked when you were looking for the picture.
I lagged out there. Ask what?
You were saying that if you had a picture, you could show us the sites, like
Oh yeah.
the attachment sites. I'm not sure what kind of picture you were looking for, but in my travels through the websites that I manage, I did come across the RCSB protein database. Are you familiar with that?
Yeah, for sure.
Oh, okay. Okay.
PDB.
PDB. Yeah.
Protein database. Yeah, no, I hopefully. Well, I don't know. Did you guys see it? Like, I was showing off the Moclo stuff. I showed some pictures of it. I mean, like the restriction sites and like how the overhangs like can bind to each other and stuff like that.
Yeah, but that's well, for me, that's all kind of Greek because protein's just like a tangled mess of lines and strings and funky.
There's a lot of that. Yeah, for sure.
But somebody understands it, and that's good.
Somebody does. Yeah, they work with it. Yep. Well, I can still link some of these videos to you guys, so you guys can always check it out on your own as well. It was yeah, definitely worth if you're keen on making this kind of stuff. Yeah, very good to look at it. I will do that in a minute, and then I think I have to go actually because I haven't eaten lunch yet, and it's like 1:30, and I'm kind of hungry.
Likewise, somebody is making bacon, and I require it.
Oh,
It's been fun, y'all. I gotta go. Thank you for your time.
Thank you
How
for
do you pronounce your handle?
a quanker,
A quanker.
a singular quanker.
A singular quanker.
Correct.
I have so many questions, but I'm going to save those for another time.
Very good.
Bye, guys.
Okay, cool.
Yep.
Bye bye. Thanks
See
for
ya.
coming, everyone. Thanks for waiting. Bye bye.